Hello everyone and welcome to the video. So today we are continuing our discussion on the central dogma of biology, and we're going to actually wrap it up today and talk about the second main process that's related with this central dogma, and that is this last step of DNA translation. So as the name implies, we will be doing some translating, almost changing languages of sorts, if you will. All right, so in DNA translation, we first need to consider what just happened right before we start this process. So, right before DNA translation, we're thinking about this mRNA message that was made by transcription. So, we talked about DNA transcription in the last video, and that mRNA that was made during DNA transcription is going to be sent into the cytoplasm, and in DNA translation it's going to be read by the ribosome to make a protein, and that's kind of shown in our picture here. We're going to break down what's happening there in just a sec. So how this is going to happen. So the ribosome is actually going to be reading 3 nucleotides of the mRNA at a time. So again, to point out what we're looking at here. So this is our mRNA strip here. It goes through. This is our ribosome in the background here, kind of this big green blob. Remember when we talked about the cell parts, we talked about the ribosome being made up of 2 halves. We have a large upper subunit and a small lower subunit. And then we'll talk about these in a sec. On our mRNA, we have our letters here, and these are representative of the RNA nucleotides that are built into our mRNA. So C, G, U, A, this is cytosine, guanine, uracil, and adenine. So when the ribosome is basically reading or decoding or translating the code that is on this mRNA, it's going to look at 3 letters at a time. And each 3-letter nucleotide combination is called a codon and codes for a certain amino acid. So this word codon here is really important. I underlined it. It's a really important vocab word. And this picture shows it here. We kind of break up our nucleotides into groups of 3, and that group of 3 is called a codon. So that is part of the reason why when we've been doing past examples with both DNA replication and DNA transcription, we've been breaking up our base pairs into groups of 3, is because those groups of 3 become really important in this last step of translation. So again, when we have groups of 3 on our mRNA, we call those codons. And when we're thinking about how this gets matched up, the amino acids are brought to the ribosome by tRNA. So remember in our video from yesterday, when we talked about the central dogma and different types of DNA, we also have this other type of RNA up here called tRNA. And remember, T stands for transfer RNA. So the job of tRNA is to bring amino acids, represented by these different colored spheres here. It's going to bring these amino acids to the ribosome so that this growing peptide chain, this growing amino acid chain can be made. This is going to turn into our protein. This chain that is growing here is going to be our final protein that we're trying to make. So again, our amino acids are being brought to the ribosome by our tRNA. And if you look closely here, you'll notice that the tRNA also has its own set of base pairs. It also has its own nucleotides built into it. However, we call those a different name. The tRNA have what we call anticodons. If you know what the prefix anti mean, it's the opposite. So an anticodon is going to be the opposite bases of a codon that matches with the mRNA codon. So again, these 2 vocab words are really important. It's important we recognize the relationship between them. So again, we find codons on our mRNA. It's labeled here on the mRNA. But when we have it on the tRNA, those are going to represent our anticodons. Because if you look closely at this picture, as these tRNAs are binding to these specific codons on the mRNA, we have matching nucleotides here. So if there's an A in our mRNA, we should have a U in our anticodon on the tRNA. If there's a G, we should have a C. If there's a U, we have an A. And so on and so forth. You'll see that the anticodon is the exact opposite of the codon from the mRNA. So that is how basically the ribosome is able to tell what amino acid gets brought, is because there's only going to be a certain combination that matches here. Only certain anticodons are going to bind to these regular codons on the mRNA, and that determines what type of amino acid gets brought to our chain here. To point it out, we see that our tRNA here is GCA for our anticodon, while this one is UGC, and these are different colored amino acids. So that means that each of these codes is going to code or translate into a different amino acid getting added into the chain. And we'll talk a little bit more specifically about how that code gets broken down in just a bit. All right, so there's going to be 3 main steps of DNA translation, and we explained most of them on that last slide, so hopefully this shouldn't be too bad. Our first step is the ribosome is going to find the tRNA with the correct anticodon to bind to the codon on the mRNA. So again, it's really important here we keep track of where do we find the codon and where do we find the anticodon. If we are looking for a codon, where are we going to look? We're going to look for codons on our mRNA. Remember, our codons are found on our mRNA, our messenger RNA, while our anticodons are found on our tRNA molecules up here. These are our tRNAs. So the ribosome is basically going to say, okay, we have AAA here, so that means we need the anticodon UUU. So this tRNA is going to come in, bind there, and then add its amino acid to the chain. The second step is that the ribosome is going to take the amino acid from the tRNA and add it to the growing protein chain. So the ribosome, basically what it's going to do, it's going to have the tRNA come in, and then it's going to basically pop the amino acid off of the top of the tRNA here. We see that there's an outgoing empty tRNA because it gave up its amino acid here, and now it's going to leave. So the ribosome is going to take the amino acid from the tRNA and add it to our growing amino acid chain. And then next, our last step is that the mRNA chain is moved up 3 letters, and then the next codon is read by the ribosome. Remember that codons are read 3 letters at a time. So once we read one, once we find the appropriate tRNA to bind here, we're going to go to our next one. So we see the next one we're looking for is UUC. The anticodon for UUC should be AAG. So we have the correct tRNA coming in to bind next to give their amino acid to this growing chain. And then this goes over and over and over again, and basically until there's a stop signal, until there's a signal in the mRNA that says, okay, we're done. You don't need to keep reading. And that's DNA translation. All right, so when we are decoding mRNA, there's a few things we want to keep in mind. We'll come to this in just a second. But when we are determining what amino acid will be put into a protein, you must match it using the codon of the mRNA. So on that last slide, we were making sure that the codon in our mRNA matched the anticodon found on our tRNA. And to the right here is a tool that scientists created. We basically figured out the code of DNA, and we were able to create this table that will tell us if we know what the codon is, we will be able to tell exactly what amino acid will get put into the chain. So this is a chart that you can use to determine the amino acid that will be put into a protein based on the codon in the mRNA sequence. So what you'll see on the side here is a list, and this is a list of all the possible amino acids that you can get put into a protein. There are 20 naturally occurring amino acids that we find, and scientists have basically mapped out, they figured out exactly what codon lines or what codon translates into which amino acid. So when you look at this chart, you start from the middle and then work your way out. And then once you work through the 3-letter codon, you will eventually end up at the appropriate amino acid. So let's give this a shot. If we had the codon, for example, GGG, we just had 3 G's in a row. Try to use this codon chart and figure out what is going to be the amino acid that we're going to find here. And then you can look at whatever abbreviation it is and look at what the actual amino acid is called. So go ahead and take a second. For GGG, what would the amino acid be? Go ahead and pause the video. All right, so if we're starting in the middle of our chart here, we start at this first G. So we start at our G, and then we look here. We now are confined to this quarter of the circle. And then our next letter was G, so we see a G here. We are now confined to this top little part of the circle. And if you can tell, it doesn't really matter what the last letter is going to be. Whether the last letter is G, A, C, or U, it is all going to code or translate into the same amino acids, which is GLY, which is glycine. So if we have GGG, that is always going to give us a glycine into our amino acid chain. So that's how we use this chart, and we're going to practice more with this in just a sec. So this is going to be very similar to what we've been doing for DNA replication and DNA transcription, except now we're going to put all the pieces together. Should be interesting. So in this step, we're going to go from DNA to RNA to protein. And hopefully we recognize we are basically enacting the central dogma here. The central dogma is this process of going from DNA to RNA to protein, and we're going to practice that now. So take the following sequence of DNA, determine the mRNA sequence, and then determine what amino acids will be put into this protein. Sorry, this part shouldn't be there. Just go ahead and ignore that. We're just going to skip down to this last step. Determine what amino acids will be put into the protein. All right, so if we have DNA here, this is going to be our DNA sequence. So what I'm going to have you do is go ahead and write this out in your notebooks. And then once you have that written out, what I'm going to ask you to do is to basically transcribe this DNA sequence into the mRNA sequence that would be created. So if we're thinking about what mRNA sequence would be created based on this DNA sequence, go ahead and write that out below. So I'll give you a second to do that. Go ahead and pause the video. All right, so if we properly put together our RNA sequence, what we should have gotten was this. So T, if we have a T in our DNA, we have an A. If we have an A in our DNA, we have a U. If we have a C in our DNA, we have a G. And if we have we actually don't have a G there. So what we should have for our full RNA sequence is A, U, G, 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 A, U, C, U, A, G, U, G, G, U, G, A. So hopefully you got this as being the RNA sequence. What I'm going to ask us to do now is to actually take this RNA sequence and to use our decoder here to figure out exactly the amino acid order that is going to be created with this RNA. So again, how we do this, you go 3 letters at a time, and you're basically going to read the 3 letters here and then look at our chart and find what is going to be the amino acid that gets put into sequence here. So let's do this first one together. So if we have an A, U, G, so we're going to go to our chart here. We have sorry. Oh, okay, sorry. I was having a little trouble reading this. Sorry for that pause. When we have A, U, G here, we start in the middle. So we start in the middle, we see A here, and then we're in this quarter of the circle. Then we look for our U, and then we have G. I was just reading this backwards for some reason. This is actually going to decode to MET, which over here we see as methionine. So what I would do is right below our DNA, you can just write out the abbreviated version. So you can write MET for methionine. And then usually you put a little dash, a little hyphen, and then you can figure out the next one. So it's GGA. You use this chart to figure it out. I'm going to ask you to pause the video and actually figure out the rest of our amino acids here. And then we'll see how you do. Alrighty. So hopefully that didn't take you too long. Didn't have too much trouble with that, but let's see how we did. So as we just talked about, our first one should be AUG, which is methionine, MET. Our next one is GGA. So again, start at the middle, GGA, which is going to give us GLY, glycine. Our next codon is UCU. So we go UCU, and then that's going to give us SER, which is serine. And then we have AAG. So we go AAG. This one's a little tricky because it can come down to 2 at the end. But if you follow AAG, this is going to give you LYS or lysine. Next is going to be UGG. UGG. This one's also a little tricky. So if you follow this closely, this one ends at TRP or tryptophan. And then our last one is UGA. So UGA. And this one is just S. And this is actually what's called our stop codon. So this is going to be a really important codon. I mentioned that when a ribosome is reading an mRNA sequence, it's going to always usually end with this stop codon so that the ribosome knows, okay, I'm done reading. I can stop making this protein. This stop codon basically allows for proteins of different lengths, and it makes sure that the ribosome doesn't accidentally go to the end of the mRNA. There's not going to be any weird mutations. This stop codon is going to be a very definitive, okay, you can stop reading now. This is the end of this protein. So this UGA pattern is going to be our stop codon. And then we will also find actually the reason that we had AUG first, this methionine. Methionine is what we call the start codon. So AUG, methionine, is going to be the amino acid we find at the beginning of every single protein. Ribosomes are going to be reading through an mRNA until they come across this AUG, and that's when they know to start reading, and that's how they get set on their 3-lettered pattern. So AUG is our start codon, and UGA is our stop codon. All right, that's all I have for you all. Thanks for listening.